Hey everybody, so this week we're going to go over seedless plants. So we're going to spend the next few weeks talking about plants. This class and next class we'll talk about plant diversity, kind of how things have changed with the different lineages of plants as we get into more recent lineages. And then later on we're going to talk about plant physiology and anatomy. But specifically for this week, we're going to talk about seedless plants, which includes nontracheophytes and seedless tracheophytes. All right, but just to keep in mind, a couple things that all plants have in common. Plants evolved from green algae, and green algae were aquatic organisms. So plants really evolved to live life in a terrestrial environment. So there was a lot of things that plants had to overcome in order to do that. A couple of key characteristics that plants have is one, they're multicellular. Now remember, multicellular means that you have many cells and all the cells need each other. You can't take one of the cells away and expect it to survive because it won't. All right, that's the difference between multicellular and colonial. All plants are also photoautotrophs, meaning they make their own food using sunlight. Something else all plants have is that they have rigid cell walls made out of cellulose and lignin. Lignin is a chemical that we mostly find in wood and bark, and cellulose is something that we find in all parts of the plant. But keep in mind, the green algae that we talked about previously also produce cellulose. So that's one of those characteristics that all organisms of the lineage Plantae have, but that includes the kingdom Plantae, which we're talking about today. Next, it's important to understand that how successful plants were. All right, once plants evolved for life on land, they really took off, and we have so much diversity in plants. And they are habitat producers. They produce habitat for other organisms to survive as well. All right, so as we go through the next few weeks, one thing you should keep in mind as we go through it is different adaptations plants have evolved in order to live on land. Now remember, there's a lot of different challenges because these came from green algae that lived in the water. Living on land has different obstacles that you need to overcome. So try to pinpoint or point out the different characteristics that plants evolved in order to do that. One last thing that we will talk about in this lecture specifically is alternation of generations. So plants have alternation of generations where they have two different adult stages. They have a 1N gamified stage, and then they have a 2N sporophyte stage. And as we go through these different lineages, you will need to be able to point out these two different stages for each of the examples that we use, as well as understand the ploidy of that particular stage. Okay, so this is just a fun fact. This is the closest known relative of plants that we can find today. So this is the phylum Terrafida, which is, again, a green algae. And remember, plants came from green algae. So this is not a plant. This is a protist because it's a green algae. However, this is as close as we can find that are related to plants. And even looking at it, you can kind of see how plants may have evolved from a common ancestor as this organism. All right, we're starting to see this branching that's green. We know it photosynthesizes. It even has stalks, which are kind of like stems that we see in plants. So we're starting to see these characteristics. Specifically, this particular example is called Cara, also known as stonewort, but it is part of the Terrafida phylum. Okay, so let's talk about challenges of growing on land. All right, this is much different than living in an aquatic environment. You have different obstacles that you need to overcome. And one of them is gravity. Now, gravity is important because when you live in water, all you need to do is float. All right, if you want to get closer to the sunlight, you float, you get to the surface of the water, and then you could photosynthesize. Plants don't have that luxury. So instead, plants have to figure out ways to grow upwards closer to the sun. And you'll see that with plants, the majority of competition between plants is who can grow higher to get to the sunlight and block out the others. This poses challenges though, because you need to be able to support yourself the taller you grow. So plants have evolved different ways to do that. Another thing that plants had to overcome is desiccation, or the loss of water. Now, when you live in an aquatic environment, this is not something you have to worry about, but living on land, you have to worry about losing water. So plants have created different adaptations in order to prevent desiccation. And finally, reproduction was a challenge. Our green algae, a lot of green algae use sperm and egg for their mode of reproduction. However, sperm is flagellated, Sperm need to be able to swim in something. So when you're in an aquatic environment, this is not a problem. Sperm can swim through the water and find the egg, and then fertilization has occurred. But you don't have this luxury on land. So this is another very important point that we're going to talk about as we go through these different lineages, because there's different strategies to overcome this lack of water. Some plants, especially the earlier plants, still use flagellated sperm, which means they can only reproduce during wet seasons. So when there's flooding, then they can release their sperm for reproduction. And then when we talk about more recent lineages, then we're going to start seeing pollen being used to bring sperm to an egg. Okay, so this is the alternation of generations in plants. We talked about this in the previous lecture, but you really need to know each one of these steps and the ploidy of each one of these steps, because during the quiz or an exam, you could be given a picture of one of these stages of the examples that we're going to talk about, and you need to know its ploidy, and you need to know which stage you're looking at. Okay, so again, alternation generation, you have two different adult stages. You have a 2N sporophyte stage, which means you have pairs of chromosomes. And then you have a 1N gametophyte stage, which means you only have one chromosome. Their chromosomes are not coming in pairs. So with sporophyte stage, you have two versions of each gene, or two alleles of each gene. Gametophyte, you only have one allele for each gene. All right, so starting with the gametophyte stage. Gametophyte is one end. Gametophytes have special tissue that we call antheridium and archegonium. There's special locations on the gametophyte that produce sperm and egg. So specifically, antheridium produces sperm and archegonium produces an egg. So just looking at antheridium and archegonium, knowing it's part of the gametophyte, what ploidy are these? Well, if it's part of the gametophyte, it must also be 1N, because the gametophyte is 1N. Now the archegonium and antheridium are going to make sperm, also 1N. So we have haploid sperm, and we also have haploid egg. Okay, so everything that we looked at so far is 1N, or haploid. When a sperm finds an egg, it's going to fertilize the egg. So for each of the chromosome locations, we have one chromosome on the sperm, and the same thing with the egg. Now, when they fuse together through fertilization, now you have pairs of these chromosomes. You have half of the chromosomes coming from the sperm, half the chromosomes coming from the egg, and you have created a 2N zygote. So that first cell that is created through fertilization, we call it zygote, and is diploid, because you have half the chromosomes coming from the sperm, half the chromosomes coming from an egg, and they pair together as a diploid organism. Now, this is a single cell. This cell, it needs to grow. So a developing zygote will split its cell through mitosis, and do this over and over again until you have multiple cells, creating an embryo. So that's the difference between an embryo and a zygote. Zygote is that first cell that's created through fertilization, and embryo is the developing organism that is made up of multiple cells. But all these cells were made through mitosis, so they're all also diploid. Because if you remember with mitosis, we end up with the same thing that we started with. We have a 2N parent cell that is split into two 2N daughter cells. And eventually this embryo is going to keep dividing through mitosis until it grows up to be the adult stage sporophyte. And again, this was all done through mitosis, which means the sporophyte also needs to be 2N, because each one of the cells that these cells came from were also 2N. Okay, so let's look at the name sporophyte. This tells you everything that you need to know about what this plant does. Whenever you see phyte, it means it's going to make. So the prefix here is sporo. So what is this going to make? Well, it's going to make spores. A sporophyte is something that makes spores. Going back to the gametophyte, the same thing can be said about the gametophyte. We have phyte meaning make, gamete meaning what it's making. So this is making gametes. Gametophyte made gametes, which is sperm and egg. Sporophyte is going to make spores. Now, specifically on sporophyte, you have a particular tissue called sporangium that is going to make these spores. So what ploidy is the sporangium, knowing it's part of the sporophyte? Well, it must be 2N, right? Because it's part of the sporophyte. Well, sporangium is going to release a cell called a spore mother cell. All right, this is the mother cell to the spores that are going to be created. And this spore mother cell is going to divide through meiosis. Now, if you remember what the product of meiosis is, You start off with a 2N parent cell. Through 2 rounds of meiosis, you eventually end up with 4 haploid cells or 1N cells. These 1N cells are spores. So these are haploid spores now that came from the spore mother cell through meiosis. Now remember the difference between spores and gametes. Spores do not fertilize each other while gametes do. All right, so these spores are going to be released into the environment, and each one of these spores individually are going to grow up to be a gamified through mitosis. So if one of these cells, these spores, divide through mitosis, you're going to have multiple cells that are all 1N, because remember, mitosis, you end with what you started with. We started with a 1N cell here, we divide through mitosis, then we have more 1N cells, and eventually it develops into be the gamified. So each one of these spores will be its own gamified somewhere in the environment. And once you're at the gamified stage, you start the whole cycle all over again. All right, so this is why we call it alternation in generations. We have a gamified stage where it's a standalone organism that is haploid, and we have a sporophyte stage, which is a standalone organism that is diploid. So one generation is haploid, the next generation is diploid, which means the third generation is haploid, and back and forth. So I encourage everybody to take the time to draw this out yourself. It's going to help you immensely understand this cycle, and we're going to see it over and over again. So it's best to have this ingrained in your memory now. So I would pause this video and go ahead and do that and kind of test yourself. Try to figure out what the poly of each one of these structures are just based on your diagram. Okay, so we're going to start going through the different lineages of plants. We're going to start with the more ancient lineages, the ones that are further that separated from evolutionary history further back in time, and then we're going to get to more recent ancestors. A trend that we're going to see with this is that the more ancient lineages, you're going to see a dominant gamified stage, meaning the gamified stage, that 1N haploid stage, is going to be bigger and last longer in this life cycle, this alternation generation, than the sporophyte stage. But as we get into more recent lineages, we're going to see a switch to a more dominant sporophyte stage, where we're going to see the sporophyte stage be bigger and last longer than the gamified. Okay, so you need to be keeping track of all the diversity that we've talked about throughout this course, because that really is the main topic of this course. We're talking about diversity here. So understanding how one thing is related to another is very important. Specifically, this is where we're focusing on our tree of life that we're talked about so far. Now, looking at this, we're going to see that we have nontracheophytes, also known as non-vascular plants, and then seedless tracheophytes, which means seedless vascular plants. The common ancestor to all of these is going to be down here, which is going to be a very ancient ancestor of plants that were an algae that could live on land. All right, before that, we're going to see a common ancestor with all these plants and green algae. So it's important to remember that plants evolved from green algae. Green algae are protists, or under the kingdom Protista, while plants are under the kingdom Plantae. But all of this constitutes the lineage Plantae. Okay, so let's talk about synapomorphies real quick. All right, the synapomorphy here that started the kingdom Plantae is an algae that could live on land. So it developed certain characteristics that allowed it to live on land. Here, the more ancient lineages of plants that branched off from these early plants have certain characteristics that are common in this ancestor. And one of them is they lack vascular tissue. Vascular tissue had not evolved yet when these lineages broke off of the plant lineage. Then later on, the synapomorphy we see is vascular tissue, which is xylem and phloem. So xylem and phloem evolved later on, and this allowed plants to really branch out and dominate the terrestrial environment. So this is the separation between nontracheophytes and tracheophytes, aka non-vascular and vascular. All right, so the two examples we're going to use for nontracheophytes are mosses and liverworts. And some characteristics that all nontracheophytes have. One, they lack vascular tissue. That's actually where this name is coming from. Non meaning doesn't have it, and tracheo, just like your trachea in your throat, means tube. So nontracheophytes are things that don't produce a tube or vascular tissue. So these do not produce xylem and phloem. Another unique characteristic to the nontracheophytes that separates them out from more recent lineages is that they don't have true roots, stems, and leaves. We're going to learn more about this later on, but roots, stems, and leaves are plant organs. And you have to think about the definition of an organ to understand this. An organ is two or more tissues coming together to achieve something in the organism. So roots, stems, and leaves, if you cut any of them in half and looked at them under a microscope, you're going to see multiple tissue layers that make up these organs. So because of that, we have different names for them. So in nontracheophytes, instead of roots, we consider them rhizoids. A rhizoid is a root-like structure that only has one tissue. And instead of leaves, we have something called a thallus, which again is like a leaf, but it's only made up of one tissue. All right, something else we see with nontracheophytes is that they're still very dependent on water for reproduction. These typically grow in wet areas to begin with and also rely on wet seasons to release sperm into the environment in hopes that the sperm can swim to an egg. And just like we just talked about, we're going to see the gametophyte stage is the dominant stage of this plant, while the sporophyte stage is very reduced and a very short life stage. And we're also going to see that sporophytes need the gametophyte to survive. So the sporophyte is going to grow on the gametophyte. Okay, so the first phylum we're going to cover under nontracheophytes is bryophyta, which is made up of neom and polytrichium. So again, these are your true mosses, all right? And they have a gametophyte dominant life stage. So when you see these out in the environment, you're actually looking at the gametophyte stage. If you took a tissue sample here and looked at the genome, you're going to see a haploid genome, meaning for every chromosome, there's only one. They don't come in pairs, which means you only have one allele for each gene. But they do have alternation of generations, and they do grow a sporophyte stage. Now here's your sporophytes, this little bell-looking structure here. This is a 2N stage of this life cycle. Now it's much smaller than the gametophyte, and it requires the gametophyte in order to grow. These are growing out of the gametophyte. They can't grow on their own, unlike the gametophyte that can grow on their own. Okay, so here's the life cycle of neom. And again, if you know your alternation generations, this is going to be much easier for you. You just need to identify the different structures at each one of the stages of alternation of generations. So let's start up here. Here we have our gametophyte stage. So this is what we are seeing grow because it's the dominant stage. Now all this tissue is 1N. With neom specifically, you have two different plants for male and female. You'll have a male gametophyte growing in one location, and you'll have a female gametophyte growing in another location. Again, both are haploid. On the male gametophyte, you're going to see antheridia, which is that tissue that is going to produce a sperm. So you have tissue somewhere on this gametophyte that's going to produce sperm. These sperm are flagellated and released into the environment during the wet season. On the female gametophyte, you're going to have Archegonia at a certain location that's going to produce an egg. So Archegonia is the tissue that is going to produce an egg, which is the cell that is going to undergo fertilization. So during the wet season, sperm, this haploid sperm, is going to swim in the environment, find an egg, and fertilize it. So what do you get when you fertilize a sperm and egg together? Well, the first stage is a zygote. And what ploidy is this zygote? Since you had a 1N sperm meet a 1N egg. Well, this zygote is going to be diploid. Now this diploid zygote is going to divide through mitosis to start growing. So now you have an embryo that is developing. Again, this embryo is 2N because it's dividing through mitosis. And this embryo is growing inside the female gametophyte from before. So this is all 1N tissue out here while you have this 2N embryo growing in it. And eventually this embryo is going to grow to be the sporophyte. So you have this sporophyte growing out of this female gametophyte. And what do sporophytes do? Well, sporophytes make spores. So this 2N sporophyte is going to create spores using sporangium, which is that tissue that is found in the sporophyte that makes the spores, through meiosis. And it's through meiosis we go from this 2N sporangium tissue to 1N spores. Each one of these spores are going to be flung out into the environment to grow into individual gametophytes. All right, so let's look at pictures of each one of these life stages. All right, so again, here is your neum. Here's your gametophyte stage. This is all one N. All right, so here's your female gametophyte stage, and here's your male gametophyte stage. It's hard to tell one is male and one is female just from looking at these pictures until you take a cross section of them and look at them under a microscope. When you look at the female gametophyte under a microscope, you will see your Archegonium, which is that tissue that is going to produce an egg. Now remember, Archegonium is one N because it's part of the gametophyte, which is one N. And then you're going to have your egg. Here's your egg right here. That is also one N. Looking at the cross section of the male gametophyte, you're going to see your antheridium, which is these tissues right here. And these are going to produce sperm. Now look at the difference in how they're organized. We have the antheridium very close to the surface of this male gametophyte. Well, the Archegonium is protected. You have these thallus leaves that are covering the Archegonium. And that has a lot to do with dispersal and how hard it is to grow an egg. Eggs are costly cells to make. They take a lot of nutrients to develop. So it's important to protect that egg from the environment. Well, sperm are cheap. You can make thousands of sperm very easily, and you want them close to the surface for easy dispersal. So when the rain seasons come, the sperm will be released into the environment, they'll swim to the egg, and then you're going to have fertilization occur. Okay, so here's your neosporophyte. You will need to know the different parts of the sporophyte. Again, this is all 2N structure because it's part of the sporophyte stage. All right, so this is broken down into two major parts. So you have your stalk or your seta, which is a stalk-like structure, 2N. And on top of the seta, you have a capsule. This is where you're going to find your sporangium inside this capsule. So if you take an even closer look at this capsule, what you're going to see is the main capsule, a calyptra, which is the tip of the capsule, an operculum, which is the lid to the capsule, almost like a cookie jar lid. And then inside the lid, you're going to see something called peristome teeth, which I'm going to show you another picture of in a second. Now, when you take a cross section of this capsule and look inside of it, that is when you're going to see your sporangium, which is the lining of this chamber, and the spores themselves. So this is thousands of spores that are inside the sporangium. Now remember, the sporangium is 2N. They have a mother cell that is going to divide into spores. And the result are these haploid spores that you have here. So these will be flung into the environment and grow into gametophytes somewhere else. Okay, so when you remove the operculum, which is this cookie jar lid that is on top of the capsule, what you're going to see underneath is something called peristome teeth. So these peristome have one job. They're there to fling the spores out into the environment. So once the operculum falls off, these teeth are going to fling all the spores out for dispersal of the spores. All right, so that's it for bryophyta and neum. The next nontracheophyte we're going to talk about is hepaticaphyta. These are your liverworts. And specifically, the genus that we're looking at is Marcantia. Now, not much has changed between hepaticaphyta and bryophyta. All right, you're still going to see a dominant gametophyte stage, you're going to see a reduced sporophyte stage, and you're going to see Archegonium and antheridium. There's only a few structures that hepaticaphyta have that neum does not. Okay, so again, here's your life cycle, your alternation generations. You just need to be able to match the specific example to the part of the life stage you're looking at. All right, so we have this 1N gametophyte stage in hepaticaphyta. But on this gametophyte, you're not going to find a specific location for Archegonium and antheridium. Instead, they're going to develop an extra structure called an antheridiophore and archegonophore, which are these kind of tree-like structures here. So an antheridiophore is going to have the antheridium near its leaves, while the archegonophore is going to have its archegonia near its leaves. If you took a cross section of these, you're going to see the antheridia near the surface of the antheridiophore, and the archegonia is going to be underneath the archegonophore. And remember, that's because with Antheridia, you want to maximize dispersal, and Archegonia, you want to protect the eggs. Again, we have flagellated sperm that are released into the environment during wet seasons, which are going to swim to the egg and fertilize the egg. And what's the first thing you get from fertilization? You get a 2N zygote. So this 2N cell is going to divide through mitosis to create multiple 2N cells, which is your embryo, which is going to continue to divide to develop to be the sporophyte. Now, this is your sporophyte of hepaticaphyta. It is this very, very small structure that is found on the female gametophyte. So this is 2N structure in here. This is 1N structure out here. Inside the sporophyte, you're going to have sporangium, which is going to produce spores through meiosis. Here's all your little spores inside the sporophyte. These are going to be flung out into the environment to grow to be gametophytes again. All right, so looking at real examples of these. Here is your Archegonophore. Archegonophores kind of look like palm trees, and that's because you want these droopy appendages in order to protect the Archegonium in the egg. So if you took a cross section of this Archegonophore, this is what you'll see. You'll see Archegonium, which are these teardrop-like structures down here with an egg inside. So again, even a closer look, you'll see the teardrop structure with an egg inside. So Archegonium with an egg. Looking at the Antheriophore, they have a more open, or they raise their appendages more, and that's because you want to maximize dispersal of sperm. And if you took a cross section of one of these, this is what it looked like. You have antheridium near the tip of the Antheriophore loaded with sperm. So when sperm finds an egg, it fertilizes the zygote. The zygote develops to be an embryo, and the embryo eventually develops to be the sporophyte stage. So here is your 2N sporophyte stage. It's just this structure right here that is growing on the Archegonophore of the female gametophyte, that parent plant. So looking at a cross section of this is what we see. So here is the sporophyte. So this is all 2N structure. In this chamber, along the walls, you're going to see sporangium. Sporangium is the tissue that's going to release that mother cell. That mother cell divides through meiosis to produce spores. And all these little red dots you see in here are the spores themselves, waiting to be released to grow to be gametophytes again. If you look even closer, you'll see extra structures called elaters. So it's these kind of corkscrew-shaped structures in here. Like we saw with peristome teeth in Neum, elaters in Mercantia are there to release the spores once the sporophyte opens. So these elaters will disperse the spores by flicking them out into the environment. All right, something unique that hepaticaphyta does that Neum does not is they have a specific type of asexual reproduction called gemme. So if you look at the parent plant, this is your gametophyte stage, what you would see out in the environment. All right, and again, these look like leaves, but they're not leaves. They're called thallus because if you took a cross section, you only see one type of tissue. You don't see multiple tissues. But on this thallus, you're going to have these cups develop. These are called gemme cups. And if you look inside this cup, or you took a cross section of this cup, this is what you'd see. Here's your cup, which is part of the thallus. So all one end because it's part of the gametophyte. And then you see these unique structures called gemme. All right, these gemme are clones of the parent plant. So these are 1N gametophytes ready to start growing again. So these gemme can be released in the environment, and you have genetically identical children growing somewhere else that are to the parent plant here. So 1N gemme will grow up to be gametophytes somewhere else that are identical to the parent gametophyte. All right, so that's it for nontracheophytes. Next, we're going to look at seedless tracheophytes. All right, so this is before seeds evolved. We're going to talk about seeded plants in future lectures. This one we're just focusing on the seedless. But here we have tracheophytes, which means they produce a trachea, or they produce tubes. And specifically, these tubes are xylem and phloem. Now, seedless tracheophytes are mostly made up of ferns and fern allies. Okay, again, some characteristic of seedless tracheophytes. They have vascular tissue. They have xylem and phloem. We'll talk more about xylem and phloem in future lectures when we talk about plant anatomy. But for right now, just know that vascular tissue is made up of these two special cells called xylem and phloem. Now, with seedless tracheophytes, they have true stems, roots, and leaves. They do not have rhizoids and they do not have thallus in the parent plant. All right, which means if you cut any of these organs in half, you're going to see multiple tissues. So these are true organs now. Also with seedless tracheophytes, we see this switch to a more dominant sporophyte stage. So the sporophyte stage is going to be bigger and last longer than the gametophyte stage. So this is a complete reversal than we saw with the nontracheophytes. And the two types we're going to talk about specifically are fern allies, which are closely related to ferns, and then true ferns themselves. All right, so the first fern ally we're going to look at is the phylum Lycophyta. And the two genus or genera that we're going to talk about is Lycopodium and Selaginella. Now, these are the plants you'd see in the environment. So based on your information that you know so far, are these sporophytes or are these gametophytes? Well, the sporophyte stage is dominant with seedless tracheophytes, so these are the sporophyte stage. So this means that this is all diploid structure. Chromosomes are coming in pairs, you have two alleles per gene. With Lycophyta, you're going to see extra structures called strobili. Strobili are simply structures that hold multiple sporangium. All right, and if you remember, sporangium is that tissue that will produce a spore. Well, a strobili is this cone-like structure. You'll have multiple sporangium. These chambers are lined with sporangium. So you have multiple chambers with sporangium in it. And inside each one of these chambers, that sporangium is going to produce spores. So you see thousands of little spores in each one of these chambers. So strobili are simply these specific structures that have multiple sporangium in them. Now, a key difference between Lycopodium and Selaginella is the type of spores they produce. So with Lycopodium, if you looked in their strobili, look at their spores, all the spores look similar. Or we call this homospores, meaning all the spores are the same. We cannot tell a male and female spore. That is not true for Selaginella. If you look at this, the spores of Selaginella, you'll see large spores, you'll see small spores. So we call this heterospory, and we could tell where the female spores are, which are going to be the large ones, and the male spores are, which are going to be the small ones. So the female spores will develop to be the female gametophyte somewhere in the environment, while the male spores, the smaller ones, will be the male gametophyte in the environment. All right, so the next phylum of the fern allies we're going to talk about is Sporophyte. So we bring up Sporophyte in this class because it has a lot of unique characteristics and kind of goes against evolution in a way. All right, specifically, the genus we're talking about is Psilotum, which is your whisk fern. All right, a couple things that these organisms do that are different than other fern allies is one, they don't have true leaves. All right, they have these photosynthetic stems instead of leaves. And another unique characteristic they have is dichotomous branching, where each branch forks into two. So you have a branch growing out that will fork into two branches, and each one of those will fork into two, and so on and so forth. This is a very old characteristic of plants, and it appears that Sporophyte has reverted back to this characteristic. So just know that it's unique to Psilotum. We don't see this much in plants. And finally, another unique structure Psilotum has is something called a synangium. A synangium is simply three sporangium fused together. So here is your synangium, which is this bulb right here. If you took a cross section of it, you would see sporangium right here, and there should be one in the back as well, but you can't see because we took a cross section, a thin cross section. So synangium are just simply three sporangium fused together, and inside the sporangium, you have all these spores. All right, the next fern ally we're going to talk about is Questum, which is your horsetail ferns. Specifically, we're looking at Questum as your genus. Again, they have some unique characteristics you should be aware of. One, they have non-photosynthetic leaves. So they have leaves, unlike Psilotum, but they're just reduced. So if you look here, here is one of your leaves right here and right here. These are non-photosynthetic, but they still exist. All the photosynthetic machinery is found in the stem itself. Another characteristic of Questum is that they have strobili, just like we saw in Lycophyta. So here's your strobulus, kind of looks like a cone. And inside the strobulus, you're going to find sporangium that produces homospores. They're homospores, meaning that all the spores look the same. You cannot tell a male gametophyte from a female gametophyte. All right, so that's it for the fern allies. A couple things that I want you to note here is, one, everything that we looked at is the sporophyte stage, which means it's two-end structures, except for the spores themselves. Those are one end since they're produced through meiosis. And also notice how we did not talk about the gametophyte stage for these organisms. And that's because they're extremely reduced. But that doesn't mean the gametophyte stage doesn't exist. We're just not discussing it here. So everything we looked at was the sporophyte stage. All right, next we're going to look at the true ferns, which are under the phylum Terrafida, also known as Pteridophyta in some camps. Okay, again with Terrafida, we have a dominant sporophyte stage and a reduced gametophyte stage. We are going to discuss the gametophyte stage in Terraphytes. I'll show you that in a second. But here's all of the sporophytes that you might see. So these are different types of ferns that exist. Again, everything you can tell or you can see out in the environment is the sporophyte stage because it's large and lasts longer, which means everything here is diploid structures. All right, ferns also have true leaves, which means these are actual organs, meaning they have two or more tissues that are working together to achieve the function of a leaf, which is usually photosynthesis. Ferns also can asexually reproduce using something called a fiddlehead. So you'll see a new frond pop up out of the fern to propagate or to have asexual reproduction. All right, so here's the life cycle of the fern. Again, you should know each one of these stages. This is alternation of generations again. You should really just matching the stage to what stage of the alternation of generation we're looking at. All right, so here we're going to start with the sporophyte stage, which is the fronds of the fern and the rhizome and roots that you see. The reason I want to start with the sporophyte here is because the sporophyte stage is dominant in ferns, unlike we saw in the nontracheophytes where the gametophyte stage was dominant. Okay, so with a mature sporophyte fern, asexual reproduction happens in two ways. One, you have this rhizome, which is an underground stem that grows horizontally. As it grows horizontally, it will have new fiddleheads pop out of it. These fiddleheads are standalone fronds that can grow on their own. All right, if you look underneath the fronds of a fern, you're going to see these little circles. They're usually circles, not always, but you're going to see these structures underneath the fern called sori. Each one in singular is soris, so each soris you're going to see underneath these ferns. If you look closely at them, you're going to see sporangium. The soris is just a group of sporangium. So looking at just one of these sporangium, it creates this kind of capsule. Again, this is all two-end structure because it's part of the sporophyte. Inside the sporangium, this is where you're going to have the mother cell release and divide through meiosis to create one-end spores. So you usually find spores housed in the sporangium as well. Now the spores are going to be released into the environment. These one-end spores will each develop to be a gametophyte. With ferns specifically, we don't have male and female gametophytes because each gametophyte has both male and female reproductive organs. So each spore is going to grow up to be the gametophyte of this fern. So a fern gametophyte is made up of two parts. It has a thallus and it has rhizoids. Now, why are we calling this a thallus and rhizoids? Well, because it's not multiple tissues. If you looked at each one of these structures, you have one type of tissue, meaning it's not a true organ. On this gametophyte, you're also going to see Archegonium and Antheridium. Typically, Antheridium is near the outskirts of the gametophyte, while Archegonium is towards the center. And that is because you want to maximize dispersal with the Antheridium of the sperm, and you want to protect the egg of the Archegonium. All right, these also use flagellated sperm, so they require water for fertilization to occur. So usually this only happens during wet seasons, but sperm will be released into the environment. They'll swim around looking for an egg somewhere, and they will fertilize that egg. Through fertilization, we have a 2N zygote that develops on the Archegonium of the gametophyte. That zygote will eventually grow to be a new sporophyte. So the new sporophyte grows out of the gametophyte. But once it is grown, it does not need the gametophyte anymore. The sporophyte can grow all by itself, and the gametophyte usually shrivels up and wastes away. So this is another difference we see between tracheophytes and the non-tracheophytes. With the tracheophytes, even though the sporophyte needed the gametophyte to initially start growing, once it grows, it does not need the gametophyte anymore. If you remember back with the non-tracheophytes, the sporophyte spent its entire life on the gametophyte. Okay, so just looking at some of these structures, here's your fern gametophyte. It is very tiny, it's almost microscopic. So to see these structures, you really need to look at it under a microscope like we have here. All right, so this is growing somewhere in the soil. This is all one-end structures because it's a gametophyte. Here you have your thallus, which is a leaf-like structure, but one tissue, and you have your rhizoids. On the thallus, near the center, you're going to find your Archegonium, which are going to produce eggs. And towards the outskirts, you're going to find the Antheridia, which are going to produce sperm. Then once the sperm fertilizes an egg, you're going to have a new sporophyte grow out of that Archegonium. And that sporophyte is 2N structures. Eventually that sporophyte grows to be a full-grown fern. And on the fronds of the fern, if you look underneath the frond, you're going to see those sori, which are groups of Sporangium. Now a new structure we're going to introduce here is something called an indusium. So with an indusium, it's simply just a blanket that covers the sporangium. So it's protecting the sporangium until they're ready to release their spores. Once the spores are developed, that indusium is going to shrivel up and eventually disappear, just exposing the sporangium of the sorus. Those sporangium that can then release their spores into the environment. All right, so here's a cross section of those sori. So here's one sorus underneath the fern's frond. So if you're looking underneath the leaf here is the sorus, which is made up of sporangia. All right, so each one of these are sporangium. Here is your indusium covering the sporangium. And if you take even a closer look at the sporangia, you're going to see this structure. So the sporangium is made up of an annulus, which is going to fling the spores out when they're ready, and it's going to have spores inside, which are all haploid. So the annulus is part of the sporangium. So what ploidy would this be? Well, the sporangium is still part of the sporophyte, which means that it is 2N, which means the annulus is 2N. And inside you have these spores that are 1N, which are developed through meiosis. So the annulus is going to fling these spores out into the environment. Each one of these spores are going to grow to be that gametophyte again that we saw in the previous slide. So those are all the different lineages of plants that we're going to talk about in this video. So we start off with the nontracheophytes, the seedless nontracheophytes, which are more ancient lineages where we have dominant gametophyte stage. The sporophyte grows on the gametophyte. And we talked about the alternation generations in each of the examples that we use, which is Marcantia and Neum, or hepaticaphyta and bryophyta if you want to talk about their phylums. Then we talked about the seedless tracheophytes, which are your fern and fern allies. And the phylums we talked about there were Solidophyta, Lycophyta, and Questum and Terrafida. With the fern allies, we just looked at the Sporophyte stage and the special structures called strobili that hold Sporangium. With the Terrafida or Terrafida, depending on which camp you're discussing this with, we looked at the entire alternation generations. We looked at the gamified stage, the dominant Sporophyte stage, and the special structures they have to produce sperm and egg or a spore. All right, so as you go through this and are taking notes, make sure you understand alternation generations and can identify each one of these stages for the examples that we gave. Also, make sure you know any of the special structures that we talked about with each one of these organisms and what they do. All right, so that's all we're going to discuss in this video.